You're listening to This Week in Cardiology from theheart.org on Medscape.
You can now access the latest in medical news on your Amazon Alexa-enabled device. Join me, Perry Wilson, every weekday morning for Medscape Medical Minute, where I highlight the top medical stories of the day. To add Medscape Medical Minute to your flash briefing, search for Medscape Medical Minute on Amazon and click Enable. Or open the Amazon Alexa app, go to Skills, search for Medscape Medical Minute, and click Enable. Then say, Alexa, what's the news? Or Alexa, what's my flash briefing? I hope you'll join us.
Hi, everyone. This is John Mandrola from theheart.org, Medscape Cardiology, and this is This Week in Cardiology for October 23rd, 2020. This week, the lessons of tocilizumab, hypertrophic cardiomyopathy, cerebral microbleeds, and surrogate endpoints. To start with COVID, in weeks past, I have said it was still bad. This week, I will say it is worse. Hospitalizations are definitely worse now. But COVID science this week offered up a huge lesson on critical appraisal of evidence. The IL-6 blocker, tocilizumab, has been proposed as a treatment for COVID-19, and it makes perfect sense that an inflammation blocker might work. On Tuesday, JAMA Internal Medicine reported a large multicenter cohort study that included 4,000 patients with COVID-19, and the study question was, is tocilizumab associated with reduced mortality? Looking back, the authors compared about 430 patients who received the drug and 3,500 patients who did not. And of course, you know, when there's a treatment group of 433 patients and a control arm of about 3,500 patients, you know it is a non-randomized observational study. So, to deal with the lack of randomization, the authors did adjustments to account for the substantial differences in baseline characteristics. They did this by using inverse probability weighting, and after said IPW, the authors wrote that, quote, baseline and severity of illness characteristics were well balanced between groups. And indeed, 29% treated with tocilizumab died versus 41% not treated with the drug. Hazard ratio 0.71, hugely significant results. This might lead one to say, well, these are big effect sizes. Tocilizumab works. But in the same journal issue of JAMA Internal Medicine were two small RCTs looking at the same thing, one from Italian authors, one from a French team. The first one, Salvarani et al., this was 126 patients with COVID pneumonia, looking at a primary endpoint of ICU admission, death, or clinical worsening of oxygenation. And again, in both groups, 17 patients had clinical worsening, no difference. Now, the trouble with this small study was that the confidence intervals of this hazard ratio of 1.05 ranged from 0.59 to 1.86. That means that tocilizumab could have had a reduction of the composite by 41% or worsened it by 86%. The second randomized controlled trial, this one from France, enrolled 131 patients. Half got tocilizumab, the other half usual care, and they used a primary endpoint that combined a 10-point clinical progression score and survival without need for ventilation. And they also used an interesting Bayesian type of analysis, and they reported that the probability of a 20% lowering of the combined endpoint was 83%, but there was no difference in mortality. 7 versus 6 patients died in each group. Okay, now to the New England Journal of Medicine RCT, which comes out 2 days after these 3 papers in JAMA Internal Medicine. A Harvard-led group enrolled 243 patients with COVID-19 pneumonia to tocilizumab versus standard care, 2-to-1 randomization, so 160 on active arm, about 80 in a control arm. And the primary endpoint was intubation or death. And what they found was 10.6% of the tocilizumab reached the primary endpoint versus 12.5% of the control. The hazard ratio was 0.83. But again, the confidence intervals range from 0.38 to 1.81. It's the same problem. The drug could have reduced the occurrence of a bad outcome or worsened it. 9 of 161 patients died in the tocilizumab arm. 3 of 82 patients died on placebo. That's hardly a positive signal for the drug. And so the conclusions from the New England Journal authors were tocilizumab was not effective for preventing intubation or death in moderately ill hospitalized patients with COVID. Some benefit or harm cannot be ruled out because the confidence intervals for efficacy comparisons were wide. So that another underpowered trial. And what's weird is that in most of cardiology, especially with our publication bias, that is the problem of favoring publication of positive studies, we most often wrestle with falsely positive studies, so-called type 1 error. These 3 small tocilizumab trials highlight the potential problem of type 2 error or falsely negative trials. On Twitter, Professors Sanjay Kaul and Frank Harrell had an interesting conversation about how big a study needs to be to feel comfortable excluding a benefit. Kaul wrote that the tocilizumab trial in the New England Journal of Medicine had an unrealistic sample size estimation. It was based on an exuberant expected effect size. He said the trial was powered to detect an implausibly large effect size of 50% reduction from a 30% placebo event rate. The actual placebo event rate was only 12.5%. Frank Harrell replied that for sample size calculations, the effect size should be based on the clinical effect one would not want to miss. So the problem is that if you say you'd like to be able to detect a 20% reduction in events, you need 4 times more patients. Having more patients in trials is not as easy as it sounds. For cost and practical reasons, trials have to make compromises. Now, one potential solution to get an answer here is to combine small trials into a meta-analysis. Turns out there's also another tocilizumab randomized controlled trial that's published on a preprint server. This was from a group at Baylor, and I'll link to it. This trial was larger. It had 452 patients randomized. Clinical status at day 28 was not statistically different or improved for tocilizumab versus placebo, and the Baylor authors found no difference in mortality at day 28 between tocilizumab and placebo, 19% in both groups. Now, on Twitter, Mike Johansson, an academic family medicine doc from Ohio, published a rough meta-analysis, and he looked at overall death, and I'll link to this. He combined the results of the 4 RCTs, 2 JAMA, 1 New England, 1 preprint, and he came up with a hazard ratio for death of 1.05. With more patients now combined, the confidence intervals got tighter to 0.75 to 1.50. The other piece of information on tocilizumab is that Roche put out a press release saying that their multinational placebo-sponsored trial of the drug, this trial called Impacta, had no statistical difference in mortality. I mention this drug because cardiologists can learn a ton from COVID-19 science, and this is classic, right? A large observational study shows an association of a drug with improved survival. The effect size is massive. But then 4 RCTs all find no benefit. Each was underpowered and could have missed an effect. But taken together, plus the press release of the coming trial, I think we can safely conclude that tocilizumab, a drug that cost approximately $3,000 per dose, offers no benefit. Next topic, hypertrophic cardiomyopathy. The combination of the pandemic and the news about SGLT2 inhibitor drugs distracted attention from an intriguing RCT of a novel agent for patients with hypertrophic obstructive cardiomyopathy. And the problem of HCM is that hypertrophy of the heart muscle, especially involving the septum, leads to high oxygen demand, diastolic filling problems, and dynamic outflow tract obstruction. Patients with HCM have reduced exercise capacity, chest pain, exertional syncope, and an increased risk of arrhythmias, both atrial and ventricular. The trial, Explorer HCM, is a placebo-controlled RCT presented at ESC and published in The Lancet, and it studied the novel oral medication called mavacamten. Mavacamten is a selective inhibitor of cardiac myosin ATPase that works by reducing actin-myosin cross-bridge formation. That reduces contractility, improves myocardial energetics. The multi-center trial, 68 centers, 13 countries, about 250 patients with HCM were enrolled. These patients had an LV alpha tract gradient of greater than 50 in class 2 to 3 symptoms, and they received mavacamten or placebo for 30 weeks. The primary endpoint was an increase in VO2 max, 1.5 milliliters per kilogram per minute, and an improvement in New York Heart Association class, or a 3 millimeters per kilogram per minute increase in VO2 without a functional class improvement. And the results were certainly positive. 37% of patients on a drug versus 17% on placebo met the primary endpoint. After 30 weeks, the post-exercise LV alpha tract gradient decreased more in the drug arm. The absolute difference between groups was about 36 millimeters of mercury. However, the mean increase in VO2 max in the mavacamten group was only 1.4 milliliters per kilogram per minute versus no change in a placebo group. 2/3 of the active arm improved one functional class, and that sounds great, but showing the power of placebo, 1/3 of those on placebo also improved a functional class. And a complete response, defined by reduction of an LVO2 gradient to less than 30 and having a New York Association class of 1, was achieved in about 27% of patients on the drug versus 1% on placebo. Serious adverse events were similar in both groups, and there were no concerning safety signals in this short-lived trial. My comments are that this is fun. Science like this makes cardiology fun. Unlike beta blockers or calcium blockers or DISO, drugs which globally act to just reduce heart rate or contractility, mavacamten is a novel drug pinpointing a more central pathophysiological issue, which is the myosin-actin interaction. And it does seem to work. If the blinding was good, the drug had modest effects on exercise tolerance. I mean, more patients on a drug achieved an improvement in exercise capacity and/or symptom relief. You didn't see a signal of adverse events. Now, you know there would be huge caveats from this, right? They screened more than 420 patients to get the 250 eligible. Most of these patients were white. The average age was nearly 60, so this is an older group. Most had New York Heart Association class 2 symptoms, so they were not that symptomatic. And a mean improvement of only 1.4 on a VO2 max is very, very modest. New York Heart Association class did not improve in about 1 in 3 patients on the drug, and nearly half of those on the drug still had gradients greater than 30. As pointed out in a nice features column from surgeons Patrick Myers and Edward Quintana, septal myectomy is much more effective in reducing gradients and has been associated with long-term symptom reduction. Of course, it is major surgery. As for long-term safety of the drug, we cannot say much from a 30-week study of a highly selected cohort. This drug is a negative ionotrope, and that should give us pause about long-term use. But I do see this as a beginning, and I agree with Myers and Quintana that if this is approved and then followed by intense marketing, and you know it will be, there could be indication creep. Indeed, these were a mildly to modestly symptomatic group of patients, and applying this data to younger patients who have more severe symptoms or gradients might be a mistake. Of course, another worry will be the high cost. Given this drug's modest benefit, what are we as a society willing to pay? The company may price its way out of overuse of the drug. The good news for patients with hypertrophic cardiomyopathy, though, is that more data is coming and other drugs in this class are being developed. And of course, the holy grail would be if this drug class improved hard clinical outcomes, such as the prevention of sudden death and heart failure. But I wouldn't count on that. That's a high bar. Next topic is brain microbleeds. Now, I visit our neuroradiologist a lot. As an AF specialist, I'm often asked to decide on antithrombotic strategies in patients who've had a stroke. I go see the patient with stroke, but my next visit is to the reading room, always in person. It's like a secret weapon for understanding what happened and what to do next. One of the things we see in the reading room often is an MRI that has cerebral microbleeds. These are radiologic markers of small vessel disease. They're present in about 1/3 of patients who've had ischemic stroke. Now, the risk of future intracranial bleeding, ICH, cranial hemorrhage, increases as the number of cerebral microbleeds increase. This, of course, raises questions about the safety of oral anticoagulants. Are you going to give an anticoagulant to someone with microbleeds? Now, in absolute terms, the risk of recurrent ischemic strokes is much higher than an ICH, but the asymmetry of bleeding in the brain is significant. The news story on this had a headline that read, "Microbleeds shouldn't influence anticoagulation after stroke." So I thought, cool, here's a study that could provide some reassurance about anticoagulation use in these patients. And JAMA Neurology published this study. And it was a sub-study of a large RCT called Navigate-ESUS. Navigate-ESUS compared rivaroxaban 15 to aspirin in patients who had had an embolic stroke of unknown source. 3,500 patients per group in that large trial. It was stopped early for futility. Rivaroxaban was no better than aspirin in these patients. But researchers had a subgroup of patients who had MRI baseline data. About half the group, or 3,700 patients, had baseline MRIs that could be assessed. And microbleeds were noted in about 11%, or nearly 400 patients of these 3,700. So you know what comes next. The researchers can then correlate the presence of microbleeds with future outcomes. And you have two groups, one that has cerebral microbleeds on the scan and the other without. And as you'd predict, having cerebral microbleeds increase the odds of bad things. Future ischemic strokes, ICH, and all-cause mortality were higher if you had cerebral microbleeds. That's not really big news. The finding that drove positive comments from the investigators, who really should know better, was this, quote, "There was no notable trend for a greater risk of ICH with rivaroxaban versus aspirin in patients with cerebral microbleeds compared with those without cerebral microbleeds." Now, here is what I almost said before I really looked at this study. I was going to say, see, cerebral microbleeds should not stop us from using DOACs. The ICH risk is increased whether on rivaroxaban or aspirin. But here's the problem. In a trial, Navigate-ESUS, of 7,000 patients, there were only 15 bleeding events, ICH events. And in this non-random sub-study, there were only 8 ICH events, 8. You can't tell anything from that. And it stuns me that journals publish this and authors draw causal inference from this. I searched the PDF of their paper and the authors mentioned low power twice. This observational analysis of 8 events gives no reliable information on the risk of ICH and DOAC use in the presence of cerebral microbleeds. Surrogate endpoints and the slender stent is the next topic and final topic. At the TCT meeting recently, I was drawn to a study comparing a novel, super low-profile stent. Now, of course, I'm not going to get into the weeds about PCI techniques. It's out of my area. What drew me to this non-inferiority study was how we interpret evidence of whether something is as good as an established comparator. So, if you have a stent that's just as good as another stent, then you want to prove that it is superior to that used now. But if this stent that you have offers some other advantage, say it can cross difficult lesions or it's easier to implant, then you can go for non-inferiority. But then you have to pick endpoints. In a trial called Optimize, the slender stent was compared with the standard drug-eluting stent. The endpoint chosen was this thing called TLF, target lesion failure. And that's a composite of things that can happen like cardiac death, target vessel MI, in this case defined by CKMB or troponin 3 times the upper limit of normal, and clinically driven target lesion revascularization, in other words, having to go re-instrument the vessel. And the rate of events was 10.3% for the new slender stent and 9.6% for the standard. And the upper bound of the confidence interval exceeded the non-inferiority margin of 3.5%, and the slender stent did not make non-inferiority. The problem here, though, there were many more events than predicted, and most of the events in both arms were due to the biomarker-sensitive definition of target vessel MI. And if you have more events than predicted, and you're using a fixed non-inferiority margin, it makes it harder to reach non-inferiority. The margins are tighter. This is unless the trial has massive numbers. Now, the PI and all the discussants each agreed that the biomarkers post-procedure were not a clinically relevant endpoint. In fact, the PI showed another analysis looking at events that were felt to be more clinically relevant, and that analysis easily showed the slender stent was not inferior. Now, why is this important? Because this is exactly how the XL trialists showed that left main stenting was equivalent to coronary bypass surgery. Namely, the XL authors included periprocedural MI, which the Noble investigators did not, and B, they used a biomarker-heavy definition that clearly biased against surgery, as surgery will always cause more enzyme release. So it's highly interesting that a group of interventional cardiologists are now bemoaning the use of enzyme-defined MI, when that is exactly how a major trial determined that stents were equivalent to surgery in left main disease. So my friends, surrogate endpoints are necessary, but you have to make sure they are clinically relevant and consistent. Death is a relevant endpoint. Stroke is a relevant endpoint. Exercise time, if it's a blinded trial, is quite good. Myocardial infarction is a legitimate endpoint if it's a real MI with clinical features. I think there is a lot less certainty with these enzyme-driven MIs after procedures, and as troponin enzyme assays become more sensitive, periprocedural MI might become even more difficult a surrogate endpoint to use. So that's it for this week in cardiology. As always, I'm grateful that you listened. And remember, if you like this podcast, it really helps if you could write us a review, give us a rating, tell a friend. This way others can find us. Until next week, this is John Mandrola from the heart.org Medscape Cardiology.
You're listening to This Week in Cardiology from the heart.org on Medscape.